Welcome to VolitionRx. This presentation describes our nucleosomics technology platform and how we intend to revolutionise cancer diagnosis. Let's consider the current state of the art. Current diagnostics consist of scanning, including mammography, CT scanning and MRI. Internal visualization techniques include endoscopy, bronchoscopy and colonoscopy. Tissue biopsy, including histochemistry, immunohistochemistry, karyotyping and genetic testing, are also carried out. Blood tests may be carried out, but currently biomarkers have poor sensitivity and specificity. Current screening for breast cancer involves X-ray based mammography. General screening has been introduced for women over 40 years of age and involves 2 X-rays per breast. The NIH recommend women over 40 are screened every 1 to 2 years, and screening has been shown to reduce death rates in women between 40 and 74 years of age. This is primarily due to earlier detection and improved treatment. Mammography is also used as a diagnostic aid following initial presentation of symptoms. In this case, multiple X-rays are taken from several angles to build up a more detailed image. The accuracy of mammography has improved since the introduction of digital imaging, which gives much higher resolution and allows easier manipulation of the images. Current screening techniques, however, are not without their limitations. The detection of small but aggressive tumours may not be early enough to allow effective treatment. False negatives occur, with around 20% of cancers missed, with potentially life-threatening delays in treatment. False positives can lead to anxiety as well as unnecessary follow-up, including surgery and chemotherapy. Overdiagnosis and treatment are also an issue. Certain conditions, notably ductal carcinoma in situ, can be identified but do not necessarily require treatment. Mammography cannot discriminate between dangerous and non-threatening disease. Finally, exposure to radiation. Whilst the dose of X-rays is low, repeat exposure is associated with a low but real level of risk. How can blood-based cancer diagnostics help? Volition's Nucleosomics platform offers simple, rapid, blood-based cancer biomarker analysis. It's easily implemented, it can be used at the point of care, and it provides high sensitivity with low false positives. High specificity also allows identification of particular cancer type. Simple blood-based screening and initial diagnosis will empower physicians. This will allow rapid referral and treatment if an issue is found, or alternatively, give peace of mind if the test is clear. Volition's nucleosomics technology is based on the detection of nucleosomes in blood. So what are nucleosomes? These are the basic structural units of our genes. They allow very long stretches of DNA to be efficiently packed into the nucleus of a cell. Nucleosomes are made up of a core of 8 proteins called histones. DNA is wrapped around this core, and the tails of the histones protrude from the core. They contain a variety of chemical modifications, and the DNA itself may also be chemically modified. When cells die, nucleosomes are released into our blood. They are normally rapidly cleared, but this process can be impaired or simply overwhelmed, leading to elevated levels in the case of disease. This occurs in cancer, but also in other circumstances, such as after a heart attack or stroke, after major trauma or surgery, and in the case of severe autoimmune diseases, such as rheumatoid arthritis. The presence of nucleosomes in multiple indications has previously limited their use in diagnostics, but they have been proposed as a way of following response to therapy or relapse of disease. Volition has solved this problem by looking specifically for nucleosomes of tumour origin. Changes to the structure of nucleosomes in our cells affect which genes are switched on or off. This is known as epigenetics. Epimutations can provide a second hit in the so-called two-hit cancer initiation model. Importantly, from a diagnostic perspective, these events can take place years before symptoms are present. It has been shown that a gradual build-up to often high nucleosome levels is seen up to the point of diagnosis with conventional methods. And we have shown that variants in the histone proteins can distinguish nucleosomes of tumour origin from those released under other circumstances. The pattern of chemical modifications is also modified in cancer nucleosomes, and importantly, between nucleosomes from different types of cancer. Volition's technology, therefore, has opened up the possibility of low-cost screening, early diagnosis, and prognosis. We can see here the principle of nucleosomics. Dying cancer cells release nucleosomes into the blood. A blood sample is taken and the nucleosomes analysed. Volition has developed a range of NuQ diagnostics based on its nucleosomics technology platform. These are robust enzyme-linked immunosorbent assays consisting of proprietary nucleosome capture antibodies which pull down the nucleosomes. These are coupled with a variety of detection antibodies which generate the cancer-specific profiles. Our dedicated team are generating the data we need to support regulatory filings to approve NuQ clinical diagnostics. Thank you for your interest in nucleosomics. To summarise, we are developing rapid, low-cost, blood-based cancer screening and diagnostic products. These will be available as manual and semi-automated kits, fully automated ELISAs for hospital use, and ultimately in a point-of-care format. Volition is now seeking collaborators with well-annotated blood samples from various cancers and stages of disease, as well as suitable control materials for further clinical studies.